Immunotherapy is, by definition, the therapy to harness the immune system as therapeutics. It utilizes your own cells and turn them into disease-fighting drugs. Our own cells are also controlled by our own genome. So we have a 3 billion base pair genome that encodes the secret of life, that governs the development, the morphology, your height, your behavior, and diseases when they go wrong. So our own cells that we utilize in immunotherapy to stimulate, to attack cancer cells, they're also governed by those genes. So in order to understand how immunotherapy works, we need to know how the genes work in order to control the immune cells. We try to understand what genes control what immune cells in what disease settings in a systems manner, which means, broadly speaking, we look at every gene, every molecule, and all the possible cell types, trying to understand what are the key problems of immunotherapy and how we can overcome them. One of the most canonical approach we utilize is unbiased genetic screens. With the technology breakthroughs such as CRISPR gene editing, we can now edit every human genes in our hands in a massive parallel fashion. So we can perturb all the 20,000 genes in your genome, or every possible base you're interested in, and then see which genes, when you perturb, would have an effect influencing the outcome of immunotherapy. And then you can develop new therapy targeting those genes that we identified. So we utilize the shotgun approach, which is in vivo high-throughput CRISPR screen, and we mutate every gene possible, and we eventually came up with a smaller number of genes that we believe truly have a profound effect on the immune cell's function. And those, if we hit them, we can potentially develop novel therapeutics. The way we develop therapeutics we call the gene editing for, of, and as immunotherapy. For the first bucket, we utilize gene editing to discover novel targets, and then we hit it with a traditional modality such as small molecule or antibodies. And the second approach is that for those quote-unquote undruggable or difficult to drug targets, we don't need to make a molecule. We can directly edit these genes inside the genome of a cell, and we utilize those cells as therapeutics. And finally, what do I mean by gene editing as immunotherapy, right? I mean, forget about the traditional approach, forget about the cells. Let's deliver the whole machinery of gene editing into the body as a gene therapy, and let that stimulate or manipulate or fine-tune the immune system so they can attack cancer or they can treat diseases. So those are the 3 buckets of how we develop new therapeutics.